Good afternoon. May I say, first of all, thank you very much for everyone joining. We're just going to hold fire for a little bit. There's quite a few people joining right now. So maybe we'll just give it a minute or two, and then it will be my pleasure to introduce Professor Russell Foster. Um, so I think we're going to make a start. We've got quite a few people still joining, but quite a few people in already. So good afternoon, everyone. Thank you very much for joining us for this, the second talk in the Meaning of Light series. And of course, a huge welcome to Professor Russell Foster. Thank you, Russell, for joining us.
Delighted.
A little bit about the series. The Meaning of Light series is about exploring light as a topic as opposed to lighting. Light has a multifaceted impact on our health and well-being, and as we seem to be becoming the indoor generation, we feel it's important that we delve into our connection to light. And maybe it's just me, but I feel that this is quite possibly one of the most exciting times for our industry as we find out more and more about how we interact with light and how we consume light. So, with that said, it gives me great pleasure to introduce Professor Russell Foster, Professor of Circadian Neuroscience at the University of Oxford, so that we can talk about light, biological rhythms, and architecture. And once again, Russell, thank you very much for joining us. Before we go in, I'm going to do a slightly shameless plug, and hopefully everyone can see this. This is Russell's book, Lifetime, a really wonderful book. If you haven't read it, I can highly recommend it. I think we might even get a chance to talk about a new book that you're writing through the talk,
Yes.
which I'm super excited about. But maybe to get us going, maybe do you want to sort of tell us a little bit about your career journey? And before, sorry, I've just forgotten one thing. What I was going to say to everyone who is joining is that please feel free to pop in questions into the Q&A section. Russell is going to present to us on his topic. So I'm sure at times we'd like to make it a little bit interactive. So don't hold your questions all the way to the end. Let's try and make it as interactive as possible. So Russell, sorry, tell us a little bit about your journey.
Okay. Well, I went to University of Bristol to study zoology because I thought I would become a marine biologist because I was a competitive swimmer and I love biology. So, the obvious thing is you do marine biology. But then in my second year, we had a laboratory practical where we recorded the light responses from a locust eye. And I just thought this was so cool. And so I then sort of went into sort of the sensory, photosensory side of things. My undergraduate project was actually recording from the frontal organ, the sort of parietal eye of a tadpole, and showing how its electrical activity changed with different light systems. That then got me into studying how birds detect day length using an extraordinary group of photoreceptors. And then that led to a real fascination with the biochemistry, the anatomy of photoreceptors. So I spent time in Holland and Germany, and then moved to the States, which is where I started the work on how do mammals, use their eyes to regulate their internal clocks. And that was a topic that I continue to work on. But what's happened is, as a result of establishing the Sleep and Circadian Neuroscience Institute, we've been able to expand into lots of different areas. So translation, fundamental science into new therapeutics, working with sleep biologists. So we're doing shift work stuff, we're doing adolescent sleep, educational programs. And so, yeah, there's a lot going on. So I feel a bit like a kid in a sweet shop, able to, I'm not entirely sure what will happen every day I come in. I mean, I have some idea, some plan, but then it all often goes in lots of different directions. So, yeah, I was in Bristol, then spent some time in Germany, Holland, then went to the States, then came back to Imperial College, and after 10 years came here, and I've been in Oxford for almost 20 years, which is extraordinary. The logic was I wanted to take our basic science and place it into a translational context. And I've got amazing colleagues here, not only basic scientists, but clinician scientist. So another job is that I head the Nuffield Laboratory of Ophthalmology, which is the research side of the eye hospital.
Amazing. I love the way you say, feeling like a kid in a sweet shop. Yeah, and I think that sort of reflects, what I was saying, that I just feel at the moment it is such an exciting time for the lighting industry to be able to learn so much about light as a subject. So maybe on that note, maybe it's time to jump into
Start.
your presentation. And just a
Okay.
little reminder to those who've just joined, actually, or note to those who've just joined, please feel free to drop in any questions into the Q&A section, and we'll be running those questions as they come up, hopefully.
But we also have a chance at the end as well,
Yes.
Eunice. Yeah, as
Yes,
well.
for sure.
Great. Okay, I'll try and share my screen. Can everybody see that? Okay.
I can certainly see it.
Okay, very good. Right. Well, absolutely delighted to join you this afternoon, and thank you, Eunice, for the opportunity of chatting. I've changed the title a little bit to Light, Biological Rhythms and Implications for Architecture. And as you very nicely plugged my book, it's not just. I'm doing it again. A lot of the background for circadian rhythms and sleep you'll find here, hopefully in an accessible form. But as Eunice already sort of said, I'm working on a new book, which I've got to finish by the end of the year for publication in January 26, which is Light, the Science of a Revolutionary Force for Health, ranging across the nature of sunlight. It's remarkable how few people actually understand how light is generated in the sun, going through evolution of photosynthesis, human vision, all through the various topics, and finishing on the built environment and optimizing light for human health. So I've written 10 and a half chapters. I still haven't done chapter 12, so I'm expecting lots of feedback from you, Eunice, on that chapter. Okay.
I'd be more than happy to help if I can.
Well, I would very much appreciate it. So, what I want, Eunice, you gave me a challenge because what to talk about and what not to talk about. So in the end, I've got quite a few topics to cover. And so I thought I'd sort of kick off with some background on the circadian system and sleep, some of the mechanisms, the nature of photoentrainment, the regulation of the clock by light, and very briefly, the discovery of this third photoreceptor system within the eye. And then why it's important, the importance of biological time, doing the right thing at the right time is critical, and then working against our circadian rhythms. So if we don't get entrainment properly, an appropriate exposure to the light-dark cycle, some of the issues that can arise. Then jumping back to photoentrainment and what are the parameters that the clock is looking for, and that's complicated. And so I'm going to go through a range of topics there. I want to touch on confusing advice, and one example I will talk about is sort of the angst you see in the popular media that if you look at a Kindle or a light emitting book before you go to bed, you're going to disrupt your sleep-wake systems. And I think those sorts of topics need unpacking because that's simply not the case. And then finish with a few slides on the future, what I think the interest by government in this whole area, and that I think with dialogue and research, as Eunice says, we're living in very exciting times. Okay, let's kick off with the background, and we'll start with something stunningly obvious, which is we sit on a planet that revolves once every 24 hours. And of course, what that does is generate the night and day cycle. These are profoundly different environmental stimuli, and that causes great complexity for life on Earth. And essentially all life has evolved a response whereby there's a period of physical inactivity, So an organism is not moving around within an environment which it's not adapted to. Sensory systems aren't adapted. And in us, that's sleep versus physical activity. And for us, that's consciousness. The two systems, sleep and consciousness, interact profoundly. And I guess the point I'd like to make is that this body clock, or the circadian system, anticipates this 24-hour light-dark cycle and adjusts and fine-tunes our physiology in advance of the changing conditions. So you see that our biology is immensely dynamic. And I just want to sort of illustrate that point in this slide here. So we see 6 different boxes. The time base for each of them is from 2 p.m. to 2 p.m. So a 24-hour time base. And I'm just going to start with just showing you melatonin as an illustration of a circadian rhythm. And we'll come back to melatonin later because it's often confused as a sleep hormone. It isn't. The reason I'm showing it is because what you see is that melatonin rises in anticipation of the night and it falls in anticipation of the dark. So it's not a driven rhythm, it's actually under the control of the circadian system, which is ticking away within our bodies. That's melatonin. We can look at a whole other range of parameters. Growth hormone is almost exclusively released during the first half of the night, which is interesting because if there are children with potentially growth hormone problems, they have to be kept in overnight, at least once, maybe 1 or 2 nights, to see what's going on with their growth hormone. But of course, growth hormone is also associated with tissue repair. So tissue repair is often occurring during the first half of the night. Core body temperature drops about 1 degree in anticipation of sleep. And indeed, if you block that drop in core body temperature, it can actually make it more difficult to go to sleep. And rises, of course, in anticipation of activity the next day, increased metabolic rate, and muscular efficiency. Cortisol rises, peaking in the morning, throwing glucose into the circulation, increasing heart rate, all associated with increased activity when we wake up and throughout the day. Testosterone is an interesting one, peaking in that morning window here. The reason is unclear, but it is associated with greater sperm motility. And then finally, and I'll touch on this, multiplication accuracy and other cognitive functions change really quite dramatically across the day. And I'll unpack that in some detail. The key point is that the circadian system delivers the right materials, the right stuff in the correct concentration to the right tissues and organs at the correct time of day. And without this time structure provided by the circadian system, our biology, including our sleep-wake cycle, is plunged into chaos. It's sort of this incredibly powerful sort of timer that cements our biology together. Now, if we think about the generation of circadian rhythms, we need to go into the brain. And here's a diagram of the human brain, and here's the pituitary gland, and here's the spinal cord, and here's the optic nerve. And this little area here is the anterior hypothalamus. And if we enlarge that area, we see sitting above what's called the optic chiasm, which is where the optic nerves go in and fuse, a paired structure. In humans, there's about 100,000 cells in total, which is really tiny, thinking about the bigger picture, when it's estimated to be 86 billion cells in the human brain. But the point is, I can take one of those cells and it has a bunch of clock genes. Those clock genes are turned on to produce clock proteins. The clock proteins form a complex and then move into the nucleus and turn their own genes off. The proteins are then broken down, and so you then get the loss of inhibition of the clock genes, and the whole cycle starts again. So you've got this negative feedback loop, and that's at the heart of the molecular clockwork. And we have an exquisite understanding of that, and I won't go into the details now, but happy to send some papers if anybody's interested. The point is that what we generate is a 24-hour oscillation in clock protein production and degradation. And this is at the heart of the molecular clock. But this clock is of absolutely no use at all unless the internal day generated by these clock genes is aligned to the external world. And that's a process called entrainment, and the most powerful entraining agent for most life on the planet is the light-dark cycle. And we know that the eye, the retina, has a direct projection called the retinohypothalamic tract, which is a small part of the optic nerve, which actually goes to the SCN, the suprachiasmatic nuclei, and sets those molecular oscillations so they're appropriately aligned. Now, that molecular rhythm is then translated into a physiological rhythm. And so I've illustrated here the electrical activity in the SCN, which is low at night, but very high during the day. And those biologists amongst you might be fascinated to know that this pattern is the same in a diurnal, day-active animal such as ourselves, and a nocturnal mouse or other nocturnal animals. So the decision to become nocturnal or diurnal is not at the level of the master clock within the brain, but it's downstream from that. You can see my zoological roots emerging there. Okay. So what we used to think is that the SCN simply forced rhythmicity on the rest of the body and was driving those rhythms that I showed you earlier. But what's turned out to be the case, and it was a surprise when it was first launched, it's not that long ago, is that probably every cell in the body has the capacity to generate a circadian rhythm. It has that molecular clockwork. And so what's happening is that the master clock in the suprachiasmatic nuclei are coordinating the rhythmic activity of billions and billions of individual cellular clocks organized through the organ systems and tissue systems of the body. So we often, I'm just as guilty as anybody else, often sort of as shorthand talk about the biological clock. But actually what we're doing is actually a circadian network, a circadian system of a whole mass of temporal time biology, all being coordinated with respect to each other. Okay.
It's quite fascinating stuff already, Russell. So I'm absorbing. I'm absorbing.
Right. I'm trying to get my... there we go.
Do you mind if I just, because there's been quite a few people joining since we started.
Yeah, sure.
And just a little reminder that if you've got any questions, please feel free to drop them into the Q&A section through the talk. But we'll also have a Q&A section at the end too. So yeah, please don't hesitate to send questions through. Sorry, Russell, back over to you.
No problem. So that was an introduction to the sort of the clock, as it were. And I've just said it's not a good idea to call it the clock, but let's call it the circadian system. And then move on to sleep. And of course, it's more than just the circadian system. We've got an extraordinary set of neuroscience here. And we now know that wake is underpinned by the wake-promoting neurons within the brain, one of which is the lateral hypothalamus, which is doing a lot. It's a tiny little structure in the hypothalamus, just in front of the suprachiasmatic nuclei, producing orexin, which then projects to all of these neurotransmitter systems. And during the wake state, our cortex, which is, all this stuff here, is being bathed in these neurotransmitters that keep us awake. During the sleep state, we've got a whole bunch of sleep-promoting neurons, two inhibitory pathways based upon galanin and GABA, which are projecting to those neurotransmitters and effectively reducing their output. And then, of course, as many of you will know, we have two sleep states, the non-REM, non-rapid eye movement, and rapid eye movement sleep, which oscillate backwards and forwards every, sort of 60 to 90 minutes. And during REM sleep, what's interesting is you have this muscular paralysis. So you really are paralyzed from the neck down under normal circumstances. Quite interesting. There are conditions where you don't get that paralysis. And so people have acted out their dreams, because this is where we have our most vivid and complicated dreams, and killed their partner quite unwittingly, and haven't been sent to prison because they've been shown to have REM behavioral disorder. The other thing is that whilst you're in REM sleep, acetylcholine is turned on, one of those neurotransmitters. So you've got this complicated yin and yang of neurotransmitter release and modulation. But what controls this timing? Well, no great surprise, the circadian system is part of it, but there's also sleep need, which is often called the homeostatic drive for sleep. And so let's look at those processes in a bit more detail. This is the classic two-process model proposed by Alex Borbley, who's still alive, and Zerjadan, who tragically died in 2018. So if we look at, in this cartoon form here, wake and sleep, we see that and we'll look at sleep pressure first. From the moment we wake up in the morning, the sleep pressure rises and rises and rises. And then while we're asleep, the sleep pressure dissipates. So the circadian system then interacts with this, and it provides essentially a circadian drive for wake. So first thing in the morning, the circadian drive for wake is relatively low because the sleep pressure, the pressure to go to sleep, is low. As we go through the day, the sleep pressure rises, but it's counteracted by a drive from the circadian system to stay awake. And whilst the circadian drive exceeds the sleep pressure, you'll be awake. And just before we fall asleep, the circadian drive for wake, interestingly, is at its highest point. The circadian system will then say, okay, it's time for sleep. The wakefulness drive diminishes. You then fall asleep. And then whilst we're asleep, there's a very low drive for wakefulness. It's probably also an active input to stay asleep. But as you approach morning, the wakefulness drive increases, exceeding the diminished homeostatic or sleep pressure, and then at this point we'll wake up. And
so Russell, I'm going to jump in there and just say,
yeah.
I'm glad you've sort of presented this sort of graphic, because it sort of explains to me why I just suddenly go to sleep at night. Yeah, as in I might be sort of sat on the sofa, maybe watching telly or a book, but clearly the circadian drive has completely left me.
Yes. Yeah, or it may well be that the circadian drive is there, but you've got so little sleep, your sleep pressure is overwhelming and has exceeded your circadian drive for sleep. So that's a problem. Okay.
Can I just stop you there? I've just spotted a couple of questions that come through the chat as opposed to the Q&A. The first question, which has come from Chris Pro, so I think it comes back to some slides earlier. So apologies, I've missed these. But I think the question will make sense. In respect to people who have no vision, do they have misalignment issues?
Absolutely, we'll talk about that absolutely. Yes, it's a big issue, and it's something we're working on. Yeah, if you've got no eyes, you fail to entrain, and I will make that point fairly shortly. Yeah.
And then there's one more question that's come through from Simon Carter. Thank you, Simon. How do circadian rhythms vary between people exposed to the same light conditions, especially for neurodivergent people?
Very good question, and it's something I'll touch upon fairly shortly when I discuss chronotype. So brilliant. Absolutely. But it's a really important issue. I mean, we've done a lot of work on sort of circadian rhythms and sleep-wake cycles in individuals with schizophrenia, for example. And I don't have those slides in this deck, but it's perhaps another talk at some point. But the sleep-wake is absolutely smashed, and it raises some very interesting issues, which I will touch upon later. Good.
Thank you.
Okay.
Thank you.
And whilst we sleep, of course, there's a whole bunch of essential biology going on. Now, what about melatonin? This is a question. I put this in with Ewis's advice, because everybody asks about melatonin. And so let's just talk about what we do know. We know that there's this retinal input to the suprachiasmatic nuclei, and then there's a multisynaptic pathway out of the brain into the spinal cord and the superior cervical ganglion, which then innervates the pineal. And then this innovation drives melatonin production. So here we see, again, a 24-hour time base. The gray area is when it's dark. And what you see, as we've seen before, is melatonin rises and diminishes. And this is a circadian rhythm in melatonin synthesis and release. But what we also know is that if you change the duration of the dark cycle, as illustrated here, then you can change the duration of melatonin release. So melatonin is really interesting. It's both influenced by a circadian rhythm, master clock in the SCN, but also this rhythm is modified by the duration of dark in the light-dark cycle.
So presumably, and maybe you're going to talk about this in a second. Presumably, in our electrified world, the reduction of the dark cycle has an impact on melatonin production as well.
It does. It certainly does. Yeah, if it's bright enough, and we will again touch on that. So I just wanted to sort of give the biology here, and of course it's that expanding and contracting melatonin pattern that regulates the reproductive responses in mammals. So the rut at this time of year is because the night length is increasing. That is encoded by melatonin, and that then interacts with the reproductive system and causes mating at this time of the year, and the calves and the ewes, sorry, the lambs will be born in the springtime when there's lots of fresh grass to sustain milk for the mum and weaning for the youngsters. So let's just sort of summarize melatonin. Melatonin is often confusingly termed as a sleep hormone, and this is very misleading. It is a mild modulator of sleep, not a sleep hormone. And I can send you a paper which summarizes all this. Melatonin is a biological marker of the dark. Melatonin is released from the pineal, peaking in the blood around 2 to 4 o'clock in the morning, then declining as you approach dawn. And that's true for both day-active, diurnal, and night-active animals. So a mouse, of course, is nocturnal, and its melatonin is high at night when it's active. So clearly, it isn't a universal sleep hormone, as it's often depicted. The duration of melatonin release parallels the duration of darkness. Individuals who do not produce melatonin, and if you've been unlucky enough to be a tetraplegic individual and you've broken your neck and you don't have that neural input to the pineal, or if you're on beta blockers, show a much greater than 80% reduction in melatonin production. But sleep continues with essentially only minor changes, emphasizing that it is not a sleep hormone. Melatonin supplementation, so your 3 milligrams of melatonin that you take to try and get to sleep, well, it does increase total sleep time by about 30 minutes, although that's the best recorded effect that has ever been documented. And it does reduce the time to get to sleep, so it reduces sleep onset latency, and that's around about 10 to 50 minutes. So it emphasizes the point that it's a modulator and not a driver of sleep. And then finally, melatonin seems to have a small direct action to promote sleep, and it provides an additional signal to tell the brain it's nighttime. So that's melatonin. I'm sure there'll be questions about melatonin later, but that's sort of thrown it in for interest. Okay. So now I want to move, if everybody's happy, to photoentrainment, the regulation of the circadian system by light, and the discovery of this third receptor system. And just to emphasize sorry, Ulysses, was that?
No, I was saying, yeah, let's go for it. Absolutely.
Okay. And so just to be clear what I mean by entrainment, we've seen this in one form earlier, which is that the molecular rhythm of clock proteins is the peak is occurring at the same time day after day after day. But going back to the question we had earlier, if you don't have any eyes or you're exposed to a weak light dark cycle, as for example, elderly individuals are within the nursing home, then what happens is the clock continues to tick, but it gets later and later in time. Most of us, 90%, have a biological clock that is longer than 24 hours. So under constant conditions, we would get up later and later and later. So how is the SCN trained by light? And of course, the familiar function of the eye is as an image forming vision detector, detecting an object against its background, either in black and white or using different colors reflected off the surfaces of objects to identify more clearly an object against its background. And the less familiar function of the eye is, of course, as a non-image forming light detector or a NIF receptor. And what essentially this other sensory task is to detect levels of light intensity. And that's now been shown to be important for the regulation of circadian rhythms, which will be the main topic of today. But also there are direct effects on sleep, alertness, mood, cognition. The brighter the light, the more alert you are. Part of our pupil constriction is based upon these receptors. There's an input, as we'll discuss, into the visual system, and it can affect metabolism and hormone release. Clearly, we've talked about melatonin, but I'll touch on serotonin and dopamine fairly shortly. So this second function is a broad irradiance brightness detecting sensory system.
Russell,
Now, yeah.
We have one quick question, sorry, and it's to do with melatonin, sorry. So this one's from Mariana. Does melatonin supplementation have a negative feedback on endogenous melatonin production, assuming the person has the ability to produce it naturally if the right inputs are given?
That's a really good question. There is some evidence that yes, if you take high levels of melatonin, then you are going to desensitize the system. And that's quite important because, although I said melatonin is not doing much, there are melatonin receptors in the master clock, which probably signal biological darkness. And that might not be such a smart thing to do. So yeah, I'm not a great fan of supplement. Of taking extra melatonin. There's a big thing, of course, going on in the States at the moment with parents giving their children gummy bears laced with melatonin, and I really don't feel that that's a good way forward. And we can discuss more about that.
Thank you.
Okay, so what I wanted to emphasize is that the responses to light of the circadian and the visual system are really fabulously different. And this, of course, is a dilemma for trying to reproduce light in the built environment. What are you going to target, the circadian system or the visual system? So the first point is that the circadian system really doesn't care where the light is coming from. So it measures brightness or irradiance. Whereas, of course, for vision to work, you're capturing light from a very, very restricted part of the visual landscape. So big differences. So you've got an irradiance versus radiance detector. The circadian system is broadly insensitive to dim light, whilst, of course, the visual system, as we'll see later, is ultra-sensitive to dim light. The circadian system is insensitive to short duration light stimuli, and I'll illustrate that later on. But of course, the visual system to work is only sensitive to short duration light stimuli. And then finally, the circadian system integrates light information over minutes and hours, whereas the visual system is integrating light information over the millisecond range. It's sort of detecting light and then forgetting it's seen it because it wants to build an image of the world and not have a blur. So the thing why I got into this, and of course it goes back to my sensory biology roots, I couldn't understand how the eye can perform both tasks, fundamentally different light detecting tasks. Okay, Now, this division in sensory tasks, image forming and non-image forming, is broadly, and I say broadly, and I will unpack that a bit later too, underpinned by different photoreceptor types. And just to summarize a good decade of my life, we showed in, oh yes, and just to orientate you, these are the visual cells, the rods and cones, the photoreceptor cells. This is the inner retina, where the first stage of light processing occurs before messages are sent to the ganglion cells, which is the first time you get an action potential, incidentally, in the eye. And these action potentials travel down the optic nerve to various target structures within the brain. Now, what we showed in a couple of back-to-back science papers is that you can eliminate all of the rods and cones. And we did this using genetic models in mice, and then actually found humans without rods and cones. And what turned out to be amazing is that in the absence of any visual responses, you get normal circadian responses. So you can be visually blind, but not clock blind. Now, if you cover the eyes, this is gone. So we're not dealing with a photoreceptor somewhere else in the body. It's in the eye. And then, of course, what these data beg the question is, well, what the hell is it if it's not the rods and cones? And it led to the discovery that a small subset of the ganglion cells are directly light sensitive. We call these photosensitive retinal ganglion cells, and they use a photopigment called melanopsin, or OPN4, nothing to do with melatonin. And I can explain why later. It just happens to be a name that was picked. And so we've got this specialized photoreceptor system.
So Russell,
yeah.
there's a few questions coming through, and
Sure.
on this particular topic. So if you don't mind, I'll fire them across.
Yeah, sure.
Luke has asked, are the non-image forming receptors in the lower half of the retina more sensitive, stroke numerous than those at the upper half?
They're broadly scattered throughout the entire retina, although in mice there does seem to be some regional specialization. Humans, it doesn't seem, you don't seem to get much of a concentration in the dorsal or ventral retina. So it's a fairly, it's not completely uniform, but it's a fairly uniform distribution.
Thank you. And then we have Daniel. It's a similar question. Hi, Daniel, by the way. You can't say hello to me, but I'm saying hello to you. Is it true that the IPRGCs containing melanopsin in the eye are more densely concentrated in the bottom half of the retina to detect blue wavelengths in the upper atmosphere?
In mice, that is entirely true. Yes. Well, there is a slight density change. What you get, interestingly, in the and I'm getting ahead of myself here, but what you get is a distribution of the cone types in the in the retina. So you have, quite rightly, the short wavelength cones in the lower ventral retina looking up at the sky. And as I'll talk about later on, we now know that the rods and cones can talk to these PRGCs. Yeah.
Fantastic. Thank you. And one last question from Joachim. Could you touch upon the IPRGC subtypes impact on vision, e.g. input to the LGN and the visual cortex?
Well, yes, of course they do. And I'll mention that fairly shortly. Yes. But yeah, they really are having a huge, there's a great projection pattern in these cells. Yeah. Okay. So what we were able to show in the mouse is to take the rodless coneless retina and then isolate it, put it in a dish, and then load it up with calcium sensitive dyes. And what you can see is that when you turn the lights on, these cells show a beautiful light response. Now, and David Burson did a different approach, but found the same thing in rats. And then Dennis Dacey, some years later, showed this in the macaque monkey. Now, what's turned out to be fascinating is that even our first studies, this first paper, showed that these responses are not the same. You can get different sorts of light responses from these PRGCs. And that we'll come back to in a moment. So what we've got is a melanopsin photopigment peaking at 480, and interestingly enough, it peaks at 480 in almost all species that have been looked at. Certainly, I think ours is about 481, mice are about 480, so they all peak in the same way, and it alludes to that question asked by an individual, say, are they used for looking at the dome of the sky? Well, I think that is very likely, because if you look at the dome of the sky at twilight, for example, then of course at the horizon the wavelengths are enriched in red-orange, but the dome of the sky, because of short wavelength scatter of particles by particles in the atmosphere, means that the dome is about 480, and so the dominant light at dawn and dusk is around about 480, which is where these receptors are maximally sensitive. Now that's we can't know whether that's the reason, but it seems likely.
And
Okay.
do you mind me asking is there any is there any sort of background, sort of or sort of ancestral connection with 480? I suppose maybe a stupid question, why 480?
Well, 480 is essentially the color of a blue sky.
Okay.
And so if you wanted to maximize light detection at dawn and dusk, particularly, then that would make sense. Interestingly, of course, 480 is also the maximum transmission spectrum of water. And people have also suggested that maybe 480 is important in aquatic organisms as well. But again, we don't know. Okay. This is going back to the question. This is an example of the sort of photosensitive net that you see of the PRGCs across the arc of the eye, and we can increase the magnification. They really are very beautiful cells. Okay, so I've talked a lot about the background. So why is all this stuff important? Which leads us to the importance of biological time. And I said earlier I'd come back to chronotype, which is whether you're a morning or an evening person or somewhere in between. And there is such a thing as larks and owls. And this is an example from Till Ronneberg's lab showing essentially the percentage of the population on this axis and whether you're a lark or an owl on this axis. So you see, here's our doves, which is about 65% of the population, broadly spanning the mid-range. Then you have early types, larks, about 10% of the population. And then later types, owls, around about 25%. So by far, the 90% of the population are either neutral, going to late, or extreme late types. And actually, if you take the extremes of chronotype, these individuals could share a bed, one getting up while the other was going to sleep. Okay. So what causes our chronotype? And there are three really critical factors. One is genetics. And we now know that subtle polymorphisms in some of those clock genes can be associated with morningness and eveningness. And it's also likely that there may well be epigenetic factors as well, whereby the expression, that not the gene sequences are being altered, but the expression of genes are being altered. This is something I'll come back to again, but when you see light exposure, so morning light advances the clock, makes you get up earlier and go to bed earlier, and evening light delays the clock. You go to bed later and get up later. And I'll give you an example of that and dig a bit deeper into that later on. And then, of course, age. So you've got your genes, you've got when you see light, and when and how old you are, and in fact, whether you're a male or female. And just to show you again some data from Till Ronneberg. So what we've got here is age on this axis and morningness to eveningness on this axis. And we're comparing males in blue, females in red. So what you see from about the age of 10 is a chronotype that's going from an earlier type to a later type on average. Women peak earlier than men, about 19 and a half on average, and they tend to be less owl-like, they have more of a morning chronotype compared to males. Males peak later, about 21 or so. And then this is separate. Males and females are different all the way through until the late 50s, 60s, where getting up and going to bed occurs at about the first time since they were around about 10. I suppose the point I would emphasize is that there's about a 2-hour difference between somebody in their late teens, early 20s, and the same person in their late 50s, early 60s. So what's it due to? Well, we think it's almost certainly the changing levels of estrogen, progesterone in women and testosterone in men. You see this sharp rise associated with puberty. Women peak earlier in puberty than men, so that fits. And then this slow change to a more morning chronotype as those hormones as they diminish as we age. Okay.
Russell, a quick question from Timo. Have proportions of chronotypes in the population changed over the decades?
That's a really good question. It's difficult to answer. I suspect there will be some shift because of the light exposure will change. The genetics are probably about the same. I think the changes associated with puberty will be broadly similar. I think puberty is a bit earlier than it used to be because of high levels of nutrition. So you might expect a slight shift due to that. So, yeah, I mean, I think a follow-on question from that is, why do humans have such a range of chronotypes? So if you look at mice, for example, a particular strain of mouse, or any other animal you care to look at, they have a very, very similar chronotype. You don't have this range of larks and owls. And this has puzzled humans, puzzled us why humans are like this. And one possibility that I think we tend to latch onto is that it may well have been really useful in early human populations to have vigilance across the entire day. So you've got some people who are late types and others who are morning types, making sure that there's no attack from another group of individuals or indeed animals. Who knows? But it is a very curious feature of human circadian organization. Okay. Yes, and of course, this can all be exaggerated by social media, gaming, and all the rest of it. Okay. So one thing is, let's again translate this knowledge into something we know about human performance, which I think is fascinating. And this is a study from University of Birmingham on athletes, and they had to perform a running task. So they classified these athletes into larks, doves, and owls. And they decided, and they looked at peak running performance. In the early chronotypes, peaked at around about noon, lunchtime. The intermediate chronotypes, the doves, about 3 o'clock in the afternoon. But the late chronotypes were peaking in their athletic performance at 7 o'clock in the evening. So one's chronotype can have a big impact if you are a sports performer, for example.
So one quick question on the chronotype piece, and from Kerim, does using HRT, such as injecting testosterone, affect this in older men?
Well, it will do somewhat. I mean, I don't know of specific studies that have looked. Certainly, hormone replacement therapy in women has an effect upon the sleep-wake cycle and the circadian system. So that's done. In fact, we've done some work showing that the master clock, the SCN, has estrogen receptors. It's also interesting that women tend to have a circadian system that is more sensitive to light than men. So, for example, women get over jet lag on average far quicker than men do. That's also, interestingly, true for both male and female mice. Female mice adapt to a shifted light dark cycle faster than male mice. But I don't know of any studies, they may exist, where testosterone replacement or supplementation has shown a change in sleep-wake timing. Wouldn't surprise me at all. That's what you'd predict from the data. Yeah.
Thank you.
Okay. So yes, your chronotype can influence at least athletic performance and probably cognitive performance too. So we go to Ben Franklin, one of the founding fathers, a genius, but this statement, early to bed and early to rise makes a man healthy, wealthy, and wise, is wrong at so many different levels. I think the only thing that I find about early chronotypes is that they're insufferably smug, and they sort of bounce in saying, "Oh, I got up at 5 o'clock." Ooh. Anyway, sorry. And I'd just like to, as a late chronotype, show you some very recent data. This is published just a few weeks ago, and in the BMJ, and they looked at 26,000 people and looked at a whole range of parameters and showed that those who stay up late and those classified as intermediate, so you've got the late types and the intermediate types, had superior cognitive function, while morning larks had the lowest scores, which is interesting. And also found that going to bed late is strongly associated with creative types, artists, authors, and musicians. I'm not sure whether scientists were in that group or not. Having said that, of course, you've still got to get your optimum sleep duration. So sleep duration, of course, is important, but it's how you align that sleep duration to your sleep-wake cycle and the light-dark cycle. And they showed that getting between 7 and 9 hours of sleep each night, those individuals perform the best. I don't come back to this later, and I'd just like to stress that I was, one of the reasons I wrote Lifetime is I was increasingly frustrated with individuals, the sergeant majors of sleep, screaming, "You must get 8 hours of sleep." Well, that's an average, and sleep is like shoe size. You wouldn't make one person have exactly the same shoe size, and it's the same thing. You wouldn't impose an average value on everybody. And the key thing is, it's something I go into in detail in Lifetime, is that you need to define for yourself what your optimum sleep duration and timing is. Okay, so let's now, yeah.
I'm sorry, there's a question, and I'm also going to say that sadly, I think I'm a morning lark, so maybe I score lower, which is a little bit disappointing. But anyway, on to the question from Daniel.
The thing is, are you insufferably smug, Euless?
I don't think so, but maybe my colleagues might disagree with that statement. But so the question from Daniel is, could you speak of the findings of the Boulder, Colorado camping study?
Yeah.
And what it might imply for lifestyle factors influencing, causing chronotypal differences.
Yes. Can I come back to that, because I talk about I talk about an allied experiment to that. I mean, Ken Wright's study is just wonderful, and we can discuss it fairly shortly, hopefully. Yeah.
Brilliant. And then just one quick one from Joachim. If sleep duration might not be the best predictor, what about sleep irregularity index?
Well, I mean, okay. So how do you know if you're getting enough sleep? Well, are you able to perform optimally during the day? Does it take you a long time to wake up? Do you need caffeinated drinks and all that stuff to keep you awake during the day? Do you notice in yourself altered behaviors? More irritable, less empathetic, doing impulsive things? Have your friends, family commented upon those things? Do you critically oversleep on free days, such as the weekend? That's all telling you you're not getting the sleep that you need. But it can vary enormously between individuals, and I think, yes, sleep irregularity might be a problem, but for many people, it isn't. One other important thing is we must distinguish between sleepiness and fatigue. So sleepiness is cured by sleep. But if you're experiencing sustained sleepiness, which we'll call fatigue, that is suggestive of an underlying health condition. And it's like long COVID, classic example, sleeping much longer, but you're still feeling fatigued. So it's always important to distinguish between fatigue and sleepiness.
Thank you.
Right. So let's now talk about cognition, and it's something we've touched on already. This is some work by Kristen Kyokin's group, and we've got an improvement in one's cognitive scores versus time of day. To begin with, I'm just going to talk about 7 a.m. to 11 p.m. And these are the sorts of tasks that individuals were asked to perform. And what you see is that in adults, this peaks at around about midday. It's a fairly broad curve over here. So, you could say between 10 and 1, and maybe even 2. And of course, this will change somewhat depending upon your chronotype. But peak performance is around about here. As we've already shown, if you're a teenager, it's about 2 hours later. And what's interesting, of course, is that traditionally the most demanding subjects, such as maths and sciences, are delivered first thing in the morning. Now, that's imposed because the teachers are coming in and they're doing quite well. They're feeling pretty cognitively alert, whereas the kids are still waking up. By the middle of the afternoon, the kids are firing on all cylinders and the teachers are declining. So we haven't optimized our teaching or indeed exams to the highest cognitive performance of teenagers. So, for example, Mary Cascadon in the States has delayed exam times to the early afternoon, and scores went up hugely. Okay, but let's now go beyond 11 p.m. And what is so clear is that around about 5 o'clock in the morning, you've got your lowest levels of cognition. Now, I think this is what many of us might experience. But what Drew Dawson did is give you a sense of how bad our performance is in these early hours of the morning. So he looked at the impact of alcohol this is clearly an Australian, impact of alcohol on cognition. Versus time of day. And what he showed is this is the decrement in cognition with alcohol when you're legally drunk at -15. But in the early hours of the morning, your drop in cognition is -20. And so the take-home message is that if you happen to be driving a car in the early hours of the morning, be aware that your ability to drive that car, on average, is going to be worse than if you consumed sufficient alcohol to make you legally drunk. And that's a big drop in cognition, which accounts for the fact that accounting for traffic volume, then this early morning window is by far the most dangerous time to drive. Okay, let's now look at something completely different, which is cancer. And this is when you should take your chemotherapy. And this is stuff that's been around for a long time. So this is an early study, only 118 children, but the data were very clear, who were given their chemotherapy either in the morning or the early evening. And then their survival over, about 6 years or so, was then taken into account. And what was very striking is that those kids that had their chemo in the evening, there was 70% survival over this time range, but those kids that had in the morning, there was 35% survival. So this is the same drug, the same concentration, but at different times, big differences in terms of efficacy. We also see similar data, and this is ovarian cancer. Again, Schedule A and B, everything was the same, except it was given morning or evening. One of the drugs was given morning or evening. And this is really quite striking, that if you look at survival over 5 years, then there was about 45% survival in Schedule A, but that dropped to 10% in Schedule B. Again, same drug, same concentration, different time, bigger effects. So time of day of your intervention really, really matters. And of course, what it relates to is the fact that cancer cells are dividing very, very rapidly. Chemotherapy is designed to kill rapidly dividing cells. And so the cancer cells are more vulnerable. But of course, those drugs will also kill healthy cells. And so ideally what you want to do is give the chemo at a time when the hair follicles, the bone marrow, blood formation, the gut and the liver are not undergoing cell division, so that you will mitigate the effects of these really rather nasty poisons.
And is that being adopted into sort of into everyday sort of clinical practice?
Broadly speaking, no.
Okay.
And in fact, it will be. It's a really interesting issue, and again, maybe we can talk about that at the end of this section. So we've talked about cognition, we've talked about cancer. Let's talk about immune function. Now, what's interesting is that the immune system is it's reduced in terms of its aggressiveness during the late part of the day and when we're asleep, but it's turned on and up and is much more aggressive during the first part of the day and during the day. Why? Well, we think it's because the immune system is turned down when it's not completely needed. So you're not moving around when you're asleep. You're not encountering new pathogens to quite the same extent statistically. And so it's probably wise to turn it up when you're going to be meeting pathogens when you're moving around during the day. So why isn't it turned on at full throttle all the time? Well, of course, one of the great problems of having a really aggressive immune system, great for killing bugs and invaders, but you can also get autoimmune problems. And so it seems to be turned down to reduce the chances of autoimmune disease. Okay, with that in mind, let's look at the effects of the influenza vaccine, for the flu vaccine H1N1 in individuals who are in their 70s, given the vaccination at two different times. That's the morning and that's the afternoon. So you see an antibody titer of 400 versus 100. So a really big effect of time of day, which entirely corresponds to what we understand of the circadian gating of the immune system. And to answer your question, Euless, is this currently being introduced into practice? No. We've been working, of course, with the COVID vaccine people here in Oxford, and they're now taking this sort of information on board to look at efficacy at different times of the day, particularly in groups that are particularly vulnerable, like the elderly.
But it seems it's not just sort of the influenza side, but obviously the cancer treatment, and it seems, something quite simple, could be adopted very easily.
Well,
and have greater efficacy, or it seems maybe that's just a simplistic
well,
way
no,
of.
I mean, half of my family are medics, and I say, why the bloody hell aren't you paying attention to this? And there's two reasons. One is they were never taught this stuff when they're doing their medical training, so they don't know about it, and they're too busy to try and dig into the literature. And the more pragmatic reason is, and they say, you don't understand. We're running as fast as we possibly can to stay where we are, trying to add a time, getting anybody in for any sort of treatment at any time it is just so difficult that we can't factor in yet another variable. I counter that by saying, well, okay, I get that. But why isn't more effort being put into developing drug pumps or timed release tablets, which could then deliver the drugs at an appropriate time? The data are consistent with the findings of less robust and stimuli later in the evening. I want to finish with this because, again, I think this is really interesting. And that's drug testing in in when you're developing drugs. And of course, quite rightly, we test our drugs in mice before they go to humans. But that has problems. Currently, drug testing is performed on nocturnal rodents and extrapolated to a diurnal species such as ourselves. So if we just do a cartoon of the circadian phase or timing of a mouse versus a human, then they are completely in antiphase. Now, there was a study done on a drug which would improve stroke outcomes. So a surgical stroke was given or inflicted on a mouse, and the drug was then used. And the individuals came in the morning and showed that the drug really did reduce the size of the stroke. So they thought, and the data was so good and so clear, they thought, right, we're going to speed this into human trials. So what they did was go in and, again, during the first part of the day, gave the drug to humans and had no effect at all. And then the penny dropped, although we've been screaming at them for years, that the drug in the mice was given at the beginning of the sleep phase, whereas the drug in the human was being given the beginning of the wake phase. So what happens when you give the drug at the beginning of the wake phase in a mouse? Doesn't work. And now in humans, it's being given later in the day, and it is working. So this really emphasizes that when we translate from mouse to human, we've got to be sensitive to the biological time, not the clock time. And there's an argument that we've missed, some wonderful drugs out there because they failed. And it could well have been because they were examined or tested at the wrong time. Okay, so to finish this section, time of day really does matter. Okay, let's move on to working against our circadian rhythms and sleep. And a good example of this is shift work, of course, but also lack of entrainment. And I'll come back to one of the questions about the health consequences of lack of entrainment. And this gives rise to sleep and circadian rhythm disruption. It's a term that we've coined because it's very difficult to disentangle what are the effects of sleep disruption versus circadian rhythm disruption. So we've lumped them together as SCARD. What are the consequences? Well, we see physiological disruption and psychosocial disruption, which interact. What do I mean by this? Well, of course, disturbances of the hypothalamo-pituitary axis, so changes in cortisol, thyroxine, reproductive hormones, growth hormone, big effects upon memory and cognition, leptin and ghrelin, these metabolic, the hunger hormones here, the satiation and the hunger hormone, and this glymphatic system, which is a recently discovered waste clearance system within the brain. And that's turning out to be really interesting, with a lot of clearance of misfolded proteins such as beta amyloid whilst we sleep. And of course, beta amyloid is associated with a higher risk of developing dementia and Alzheimer's. And there is, again, an argument and some data which supports it, which is poor sleep-wake during the middle years can be a risk factor for dementia in later years. And it could well be because of this glymphatic system. And it's been shown that even one night of no sleep, you can actually detect increased levels of beta amyloid within the cerebral spinal fluid. So there's a whole bunch of physiology. But also you've got the psychosocial. So you realize you're not functioning optimally. So you have difficulty coping. That can lead to low self-esteem, a whole bunch of social issues about how you interact with others, increased frustration and anger, increased worry, leading into anxiety and depression. And of course, these two systems interact, providing big changes in our emotional, cognitive, and physiological behaviors. So let's just, oh yes, and the point is, of course, that these will feed back on SCARD, making it worse, so you can fall into a really dangerous positive feedback loop. Okay, so let's look at these in a bit more detail. And So if we think about our emotional, cognitive, and physiological health, we see with SCARD increased fluctuations in mood, irritability, anxiety, loss of empathy, the failure to pick up the social signals from others, risk-taking and impulsivity, doing stupid and unreflective things that you wouldn't do if you didn't have SCARD. Negative salience is really interesting. Some very nice studies have shown that the tired brain remembers more readily negative experiences and forgets the positive ones. So one's whole worldview is biased towards negative. There's a greater use of stimulants and sedatives, which can bleed into illegal drug use. We think of cognitive responses, well, overall cognitive performance goes down. The ability to multitask. What I mean by that is that we are bombarded with information, and what we need to do is select what we need to attend to the most urgently and the most effectively. And that falls apart if you have SCARD. Memory consolidation, attention, concentration, communication, decision making, all of these other parameters fall apart. And I will show a few examples of this. Long-term SCARD, the sort of thing that you're seeing in night shift workers, but you also see it actually in the business sector as well, with very shortened and disrupted sleep. So daytime sleepiness, microsleeps, wonderful study or interesting study on junior doctors, showing that 57% had either had a crash or a near miss driving home after the night shift. Increased cardiovascular disease, altered stress responses, which probably underpins lower immunity and greater risk of cancer. The higher rates of cancer you find in long-term night shift workers, the data are now so convincing that the World Health Organization has designated night shift work as a probable carcinogen. Metabolic abnormalities, type 2 diabetes, really high in night shift workers and individuals getting little and disrupted sleep. Depression and psychosis, which I'll touch on, and I've already mentioned dementia briefly. Okay, let's look at SCARD and problem solving. And this is one of my favorite studies by Jan Born's group in Germany. And what he did is devise a really quite complicated problem solving task. And he introduced it to one group, this group here, in the morning. So they became familiar with the task and then had to do the task in the afternoon, the same afternoon. And you see that only 20% of the group succeeded in solving the problem. The second group were introduced to the task in the morning, became familiar with it, but performed it the next afternoon, but they were deprived of sleep. And only 20% of the group solved the task, nicely accounting for fatigue. They were no worse than this group here. Of course, the interesting group is introduced to the task in the morning, became familiar with it, performed it the following afternoon with a night of sleep. 60 to 70% of the group solved the task. And I think this illustrates beautifully that if you want to come up with innovative solutions to complex problems, a night of sleep can enormously enhance your capacity to do that. If we look at emotional processing, then this is a nice study from Stickold's group. And what he did was look at one group who slept normally the night before the testing, and another group who were deprived of a night of sleep, and so hadn't slept for 36 hours before they undergoing the tests. And the tests were very simple. They had to remember words, but words with a different context. So positive words, love, joy, happiness; negative, murder, hate, crime; or neutral words. And in this case, one of the neutral words was cotton. So if we look at the data where all the words are lumped together, then you clearly see that there's a reduction in the ability to remember words with one night of no sleep. But let's divide it up into the different word content. Neutral, there was a slight drop in those who were sleep deprived, but it wasn't significant. Negative words, again, slight drop, but not significant. The really extraordinary data is that those individuals who had not slept for a night had a huge failure to remember words with a positive content. So reinforcing this business of negative salience, and that one's worldview is very much influenced by the state of tiredness of the brain.
Does that tie in with empathy, do you think? Is that
Absolutely. Yeah, empathy, I mean, is again something that falls apart. And this is why one needs to listen to your friends, colleagues, and family, telling you, you're a different person. You need to pay attention. So we talked about those criteria earlier about, are you getting enough sleep? But listening to one's friends and family honestly about whether there are mood changes or changes in empathy are really important. So we're coming to mental illness, which is, I think somebody asked about earlier. And this is one of the studies that we've been very much involved with. So the majority of individuals with a diagnosis of mental health problems also experience some form of clinically significant sleep disruption. And the work that we've done in schizophrenia, I was completely shocked at how appalling the sleep-wake profiles of these individuals were. Okay, we came up with a hypothesis that sleep and circadian rhythm disruption and mental illness are linked. Now, remember I talked about all of those neurotransmitter systems at the beginning of this presentation. And so our argument was that if there's a change and we argued that there are common and overlapping neurotransmitter systems between mental illness and SCARD, or between mental health and sleep. And of course, if there's a change in one of those neurotransmitters, or more than one, that impacts upon one's level of mental health, then it's almost certainly going to have an impact upon your sleep and circadian alignment and overall functioning, your SCARD. Okay, and that sort of accounts for the fact that there are genes that have been linked for both mental illness and SCARD, and it's again some work that we have done. But it's more complicated than that, because of course the SCARD, through its short and long-term impact on those parameters we just discussed, emotional, cognitive, and physiological health, they're going to make the mental illness worse. And of course, the mental illness has the capacity to make the SCARD worse. You've got that psychosocial, physiological stress, and maybe even the impact of medications. So you've got yet another set of feedback loops whereby a slight distortion, with a change in a neurotransmitter, can rapidly, because of this feedback loop here, get worse and worse. And so what that made us realize is that we can think of SCARD as a therapeutic target in mental illness. And to cut a long story short, we worked with Dan Freeman on this paper published quite a few years ago now, 2017, and identified individuals with insomnia, with SCARD, and exhibiting levels of paranoia and hallucinatory experiences. So they were on the psychosis spectrum. And what the intervention was a partial stabilization of sleep-wake, the reduction of SCARD. So this is the control group. There was no intervention, and you see that after 10 and 22 weeks, there was no change in the level, as illustrated here, of paranoia. But the sleep intervention group, this is again the level of paranoia in the experimental group, which is the same as the control group. But after partial sleep stabilization, you saw a very big reduction, or very statistically significant reduction, in levels of paranoia. And I think this is really exciting and it's now actually getting some interest in psychiatry, that we've got some ways of a new therapeutic target for mental illness. So I think it's an exciting area. Okay, and just to finish this section on light and depression. So this is an experiment in non-seasonal depression. And we've got weeks across here, week 8 out here, and we've got a reduction in levels of depression. This is what you see with the placebo, which is interesting. And in fact, there's a big placebo effect in mental health. This is the impact of fluoxetine, which is Prozac. And what's fascinating is it takes 8 weeks before Prozac is statistically different from placebo. Then there was a light intervention, which was 10,000 lux from a light box for 30 minutes in the morning. And this is every day. And you see that by 2 weeks, light was more effective than placebo and Prozac. And by 8 weeks, light was significantly different from Prozac. So really very clear demonstration of the importance of light, morning light in this case, on non-seasonal depression. Very interestingly, if you combined Prozac and light, it looked like the effects were additive, suggesting that light and Prozac might attack, as it were, depression using different pathways.
So jumping back onto that, essentially there's a fairly effective treatment which is very cost effective.
Yeah, I mean, and it's amazing that it's not more widely adopted. And certainly my recommendation would be to try light first. One of the problems actually with Prozac is that it takes, as you see, 8 weeks to kick in effectively. And people are on these drugs and they think, my God, it's not changing, it's not getting any better. And there's a record of suicide of people who are already on Prozac, but it hasn't made a difference. And they sort of think, oh my God, I can't stand this any longer, which is really sad. And they're not told that it'll take at least 8 weeks before you begin to sense an effect. Anyway, so now we go back. I've spent a long time talking about, the circadian system, the importance of biological time, and what happens if we don't get decent entrainment and we work against, in a sense, our biological rhythms, our circadian rhythms and sleep. Now I want to move to photoentrainment and the light parameters needed, which becomes of, I think, more direct interest to architects. This is something you'll be familiar with. Light exposure has changed profoundly over the past, well, since 1800, over the past 200 years or so. In 1800, more than 95% of the population were outside as agricultural workers. This has dropped precipitously, and the estimates vary from about 1% to, as illustrated here, 10% of individuals who are spending most of their time outside compared to most people who are indoors. It's a big shift. Yeah,
I think the studies I've seen is that over here, sort of in Europe, we're about sort of 90% indoors. The Americans are 93% of their time indoors. But then when you look at the data on age groups, you've got age groups like the 18 to 24-year-old age group that seem to be spending maybe 15 to 30 minutes a day outside, and potentially if we look, if we go back to your previous slides about sort of anxiety and depression, maybe that's a big player.
Well, absolutely. Yeah, absolutely. And while we're talking about light exposure, of course, 90% of kids in Southeast Asia have myopia and need glasses. And there's some very interesting data showing that outside light exposure is a really important factor in the development of myopia. That's another story. Okay, and of course this will be familiar again. We live in modern caves, and it's really and so what I often do is get people to download a lux meter onto their smartphone and just sort of move around their environment, and they'll see maybe 3,000 lux at a window, but the time they've moved into the middle of the room, it'd be lucky to get 50 lux. In the workplace, it varies a lot, of course, as you will know, but on average 300 to 600 lux, and 600 tends to be on the bright side, rarer. And of course in places like the tubes who are under dim light as well. So the question is, well, we've got attenuation of light inside. So how to achieve entrainment in the urban environment? And it's complicated, but I think this is an important point. The problem hasn't been solved, and so we need to address it. So what do we need? The critical factors that are integrated for circadian and sleep regulation in humans are these: the light intensity, the duration of exposure, lots of individual variation, the wavelength or the color, the non-image forming light detection task, the time of light exposure, which I said I'd come back to, your age, young versus old, and indeed there's an impact of sex. I mentioned that women tend to be more sensitive to light than men, and I won't have time to go into this angle of view light history, but I will show you an example of that, and then of course the seasons. So let's think about light intensity. And it's worth flagging up that until this paper in 1987, humans were not thought to be to have their circadian system entrained by light. It was thought to be social cues, that when you ate and how you socialized. And that's because the experimenters hadn't used bright light. And so finally, the Honmas got entrainment when they used a light dark cycle of 8 hours of light, 16 hours of darkness of 5,000 lux, and then they entrained. If we look at this, and again, this is something you'll be very familiar with, and this is sort of the range of natural light exposure from 100,000 lux in direct sunlight all the way through to a quarter moon at 0.01 lux. Domestic lighting, again, somewhere in the 50 to 400 lux range. Precision engineering around about 3,000, at a workbench, 3,000 lux. So the rods are giving us our dim light vision, kicking in low levels and saturating at about 100 lux. Our cones kicking in, they overlap with the rods at around about 10 lux and saturating up to 100,000. Those photosensitive retinal ganglion cells, we've got them here kicking in at about 100 lux or so, and then not saturating out, similar to the cones in that they can they're not saturated by very bright light. But, and that's why this asterisk is here. This comparison of intensity or radiance between the rods, cones and the PRGCs is not really appropriate because it doesn't take into account light exposure duration. So you get those responses from the rods and cones in milliseconds, but it can take minutes or even hours to get response sensitivity from the PRGCs. And I'll unpack that a little bit more now. So you've got this very lovely study which brought together lots of studies. Interestingly, largely changes in melatonin, and I'll come back to that later. But you see here from this graph that the threshold for the circadian system using melatonin suppression or shifting the circadian rhythm in melatonin is around about 50 to 100 lux. And so this is part of the argument that you'll hear, that actually dim light in the evening, let's say 90 lux in the evening, is sufficient to shift the biological clock. What's not sort of stated so clearly is that yes, you can get the low light sensitivity, but you need 6.5 hours of light exposure to get that, and often much longer. So you can increase sensitivity, but having massively long durations. And it goes back to the start of the talk where I was talking about the different parameters.
So Joachim has just asked a quick question. The study from Brown here. Do you think this time the time taken of the Zeitgeber does it take that sort of daylight into consideration?
Absolutely, and I think that this is why it's a lovely paper, but it's been picked up upon and say, oh yeah, well, dim light in the evening is going to be really disruptive on our clocks, and I'll show you an example of that in a moment. And so I think some entirely inappropriate suggestions have emerged, like you shouldn't see more than 50 lux for the last 2 hours before you go to bed. Well, actually, I think this is all based upon laboratory studies, and I'll show you some other work later on. And so the whole theme of this section is urging caution, and actually we need better data, but I'll get to that. If we think of individual variation too, so this is a lovely study from Phillips and colleagues in Australia. And this shows, this is in melatonin suppression, a 50-fold difference between those individuals who are most sensitive and least sensitive to light. So there's an awful lot of variation in humans. And there may be a partial explanation for that, which I'll come to shortly. The wavelength, the color of light. Well, we thought that the photosensitive retinal ganglion cells and the rods and cones were sort of kind of isolated. They were two different systems, if and nif, that didn't really talk to each other. We got that wrong, and we now know that the cones can talk directly to the photosensitive retinal ganglion cells, and the rods have a way in too. There's a rod bipolar that speaks to an amacrine cell that then speaks to a cone bipolar, and we know that that can have an effect upon the PRGCs. Yet there's a misplaced obsession with blue light. And that's because, of course, the PRGCs are maximally sensitive at 480 nanometers. However, if you add the spectral responses of the human visual pigments, the rod, the short wavelength cone, the medium wavelength cone and the long wavelength cone, you see there's a very high degree of overlap. So you'll read many papers saying, oh, I used, blue light at 480, which would only stimulate the PRGCs. Well, absolutely not. So it could have a big effect on particularly the short wavelength cone and the rod and the medium wavelength cone. The other point I would like to stress is that we don't know precisely how the rods and cones are interacting with the PRGCs. We know that for them to have an influence on the clock, they have to go through the PRGCs. That's the way in to the suprachiasmatic nucleus. But the point I'm trying to make is that potentially there's a very broad spectral input into the circadian system. It's not just blue light. And furthermore, the models that exist at the moment are essentially additive. And yet we know from Dennis Stacey's work in the macaque that the SWS, the short wavelength cone, is inhibitory. So it's very difficult to make assumptions about the wavelength and the color of light. In some sense, it's probably safer just to use white light rather than to try and optimize PRGC light detection. Because you're going to get an input from the rods and cones as well. The task, and this goes back to an earlier question. So the first point is that we now know that there are at least 5 and probably 6 different types of melanopsin expressing ganglion cell, and they have a huge projection to the brain. So we've got the suprachiasmatic nuclei, but also the ventrolateral preoptic nuclei, the lateral hypothalamus, the subparaventricular zone, doing all of this kind of stuff. Oh yes, and critically the olivary pretectal nuclei. So part of, as I said earlier, the pupil reflex is mediated by these receptors. There's also direct and indirect pathways regulating the dopamine and serotonergic pathways. There's also, and I think this was one of the questions, modulation of the visual systems, because there are projections to the lateral geniculate nuclei and indeed the superior colliculus. Precisely how these irradiance detectors are modulating vision is an area of very active research. We also, and of course, you've got the medial amygdala, which is very much involved with mood. You've also got an input into the sympathetic nervous system, and of course, the regulation of pineal melatonin and a host of other things. So the assumption has been that these different responses to light will show the same sensitivities and dynamics. And we know that that is not the case. And so we've got to be careful about the sorts of assays of the effects of these non-image forming photoreceptors are having. The time of light exposure, which I said I'd come back to, this is really important. So we know, and this goes back to the Ken Wright experiment in the Colorado mountains, we know that dusk light And what I've got here is a light-dark cycle, and this is a sort of a daily sleep pattern in cartoon form. But dusk light delays the clock. So you go to bed sleeper and you get up later. So you go to bed later and get up later. Morning light is the opposite. It makes you go to bed earlier and get up earlier. So when we're all agricultural workers, we were getting a symmetrical exposure to dawn and dusk, which allowed us to sort of stay on cue. But we've done some interesting studies on university students. Oh yes, so getting light exposure at the right time is critical. And we looked at a bunch of university students, and a significant number were sleeping through morning light. So they were not getting the morning effects of light, which would advance the clock, make them get up earlier. But they were getting evening or dusk light, which would, of course, delay the clock. And what we found is that the later the chronotype of individuals, the less morning light they got, but the more dusk light they got. And a very similar, and what Ken Wright did was to take university students out to the Colorado mountains and compared their sleep-wake timing in their normal environment versus when they were only exposed to natural light. And what he found is just like this, is that they got up earlier and went to bed earlier. They were much more closely aligned to the natural light-dark cycle. So when you see light is important, and more evening light is associated with the later they went to bed. Okay.
So Joachim's just asked, do these inputs mean that we need to reconsider things like glare with the role of the IPRGCs and the pupil light reflex?
I'm not sure I fully understand the question. Sorry.
So maybe we'll move on, and Joachim, if you can maybe rephrase it, that would be amazing.
That would be good. Okay. Sorry, I'm being obtuse. Right. So let's now look at age and some work we did on the mouse. And what you've got here is the suprachiasmatic nucleus here in a couple of mice, and the projections from the eye to the suprachiasmatic nucleus in mice that are young versus old. And you see a really big difference in light-induced gene expression in the master clock, much more in young versus old. And these and this shows the projection, which is diminished in the old mice as well. And these are consistent with human findings, showing that older adults, healthy older adults, have a reduced sensitivity to light. It's about I think it's not huge, but it is a significant difference. So those are some of the critical factors, and I've given you some data to support that. I haven't had chance to talk, and maybe Jürgen was alluding to this, angle of view. I will talk about light history, but I won't talk about seasons, but these are all additional factors. I want to talk about some confusing advice, which kind of sort of feeds into that previous section. And some of you may remember the sort of banner headlines from ebooks, Kindle's Damaged Sleep and Health, Doctors Warn. And I think it's very important to look at those studies very carefully. So the experiment, essentially, the individuals before the experiment were maintained inside and exposed to around about 90 lux at the angle of gaze. One group was asked to read a book illuminated by a bedside lamp. Another was asked to read an ebook, a Kindle, on maximum intensity, which is around about 32 lux. Now, both read for 4 hours between 6 p.m. and 10 p.m. prior to sleep, or prior to wanting to go to sleep, for 5 consecutive evenings. So it's quite a long duration exposure. Now, if you look at melatonin suppression, there was a very clear suppression of melatonin by looking at the ebook. There was also a very clear shift in the circadian rhythm of melatonin timing. So yes, there's a significant impact upon melatonin suppression and circadian timing. But if you looked at sleep-wake timing, there was barely an effect. So there was a 9 minutes delay, or rather it took 9 minutes to fall asleep if you, after 5 nights of 4 hours of Kindle use. So this was just significant after 5 days. So a minor impact upon sleep timing. Okay, so the first thing is there's a difference between melatonin and sleep-wake behavior. So using melatonin as a surrogate measure for sleep-wake behavior is complicated. And when these experiments were essentially repeated, but instead of keeping people at 90 lux prior to the experiments, they were given 6.5 hours of 500 to 600 lux. And any effects on melatonin and sleep-wake behavior were completely abolished. And so what seems to be happening is that if you're kept under dim light during the day, you seem to sensitize the clock in the evening and make it more vulnerable to being shifted. So yes, it's conceivable that a Kindle will shift your clock very, very minorly if you are kept under dim light during the day. But if you get even relatively moderate levels of light, those effects are abolished. So it's really important to get bright light during the day. So using it, yeah.
So I've got two questions that come through from Kevin Shaw. And it actually relates to the previous section, so apologies for disrupting the flow there. Is the SWS not much more sensitive than the other receptors?
I'm sorry, what was that? Is the
SWS not much more sensitive than the other receptors?
The short wavelength cone?
Yes.
You say. Is it? Well, it will be at short wavelengths. But its dynamic range is similar to the PRGCs, but obviously it's less sensitive to the rods. But are the short wavelength cones inherently more sensitive? Well, no, unless they're given short wavelength light exposure.
Okay, thank you. And then there's one more from Kevin, which was again from the previous section. Looking at so the question is, how does this work when you have the radically different daylight time in northern latitude? So again, I think this was to do with the sleep-wake cycle and the morning and evening light.
Yeah, well, of course, it's a real issue, and what happens in having visited Tromsø, many families, they have a light box room where they go into. It's very bright light, and that's where they have their breakfast every morning. So they during the 2 months of darkness in winter, and of course then they use blackout blinds during the summer. So yes, interestingly enough, Arctic animals have been looked at. So Arctic reindeer, for example, turn off their clocks during the 2 months of constant light and the 2 months of constant dark. There's nothing to entrain to. And indeed, having a clock is of no help. So during the winter months, you feed whenever the weather conditions permit. And during the summer months, you feed all the time, so you can pack on enough fat to survive the winter. So animals, that have been in the Arctic for some time have adapted by turning off their circadian system. That's true for the Arctic reindeer, and also I think true for the Arctic ptarmigan as well.
I think the reindeer, if I'm not wrong, have some ability to change their eyes.
They do. Yes. Well, of course, you've got this incredible amount of short wavelength reflectance off of the snow surface, and so what you want to do is protect the eye, the retina, from that. And so they deposit yellow pigments, which absorbs more of the short wavelength light. Yeah.
And then thank you very much for that. And Joachim's just responded. So normally glare is only described through brightness within a visual response. However, with potential impacts from IPRGC, the visual cortex, and the sustained pupil light reflex, would this mean that IPRGC input could be important to understand, for example, glare better?
Yes, absolutely. And in fact, also, I think PRGC, understanding the PRGCs could well be very important in color constancy, which is this phenomenon whereby the color of an object stays the same under different levels of illumination, which I think is, I'm pretty convinced the PRGCs are inputting into that. But just extending this discussion a bit. Oh yes, let me just summarize the ebooks.
Yes, sorry.
So small impact upon sleep-wake timing, significant impact on melatonin suppression, and significant impact upon melatonin entrainment, the rhythm of melatonin, but effects depend upon daytime light exposure. So melatonin does not correlate with sleep-wake timing. So you've got to be really careful about how we interpret these data. Melatonin and sleep timing, different sensitivities to light. Light history modulates very profoundly evening light on melatonin, and of course behavior. And so I think I would argue you need to get good light exposure during the day, not just setting the clock at dawn and dusk. Okay, computer screen efflux. As many of you will be aware, there are computer programs, efflux, for example, or night shift, that shift the blue content of a screen to a more longer wavelength, a red enriched screen. Do they work? No. So there's no data supporting the fact that efflux has any different effects at all. And this was a reasonably recent paper I thought I'd draw your attention to because it had good numbers. And it looked at this light shift program, the night shift program. And the quote is, in the whole sample, there were no differences across the three groups. Night shift is not superior to using your phone without night shift or even using no phone at all. So a lot of this changing the spectral composition is also somewhat misleading. Okay, so I'm just going to finish up with the last few slides. The future. Well, what do we need? Well, we need information about this. And the evidence base, I would argue, for guidelines regarding human light exposure to optimize health is inadequate and can be misleading. What we need to know, of course, is this. How are we going to get it? Well, we should look at the sleep-wake timing, not just melatonin measures. We need to correlate that with alertness, cognition, and mood, and overall well-being. And we've got to get out of the lab and into the real world. And this is all doable. There are now, devices which can measure sleep-wake timing, alertness, mood, with smartphones and a quick sort of reaction time, for example, very easily. And we can do that in the real world. We don't have to sort of restrict it to the lab. Now, I was involved in this. I was the science advisor for this House of Lords report last year. And it's a fascinating experience, actually. This is on the neglected pollutants, the effects of artificial light and noise on human health. And what's exciting is that this is a statement from this House of Lords report. And if you're interested, you can just download it from the web. The government should commission research to establish how light intensity, wavelength, duration, time of exposure, light history, and age affect the circadian system. This should move beyond laboratory-based studies and investigate more realistic light exposure patterns for humans. So this actually was flagged up. I mean, I do take some responsibility for this. And what's also turned out to be exciting is that one arm of government, which is, I'm sure some of you will be familiar with, COMAIR, the Committee on Medical Aspects of Radiation in the Environment, So this group advises on the health effects of natural and human-made radiation, both ionizing and non-ionizing. And they're very interested to commission a report of what we know and, more importantly, what we don't know and what experiments need to be undertaken to try and get the information that we've been discussing this evening or the late afternoon. Okay, so we've spent quite a lot of time. Been great questions. I hope that the background on the circadian and sleep systems and the photoentrainment pathway was useful. And also flagging up the importance of biological time. Really, I can't emphasize enough about what you do when matters hugely, whether it's cognition, whether it's taking medications or athletic performance. Everything is being gated by this internal timer. The consequences of not getting good entrainment and working against our circadian rhythms of sleep. And then the parameters, the light parameters that we need to understand to, in a sense, if we're going to be inside, then we need to try and create a sort of a light environment or an architecture which optimizes human health. We don't have all the answers at the moment, and I suppose the default would then be, well, we just need to mimic natural light as much as we can from the outside and bring it in. We need to deal with some confusing advice. I'm not sure that the arguments have been entirely coherent. And so basing guidelines on lab-based experiments is, I think, has been too early, and we need to think more carefully about that advice. And then finally, I'm very optimistic. I think that we are in a position where people like yourselves are really interested in this whole topic of light and want to use the emerging information. And I think that the government will commission research to try and identify some of the missing bits of information that can make the design of our buildings and built spaces more optimal for human health and well-being. And with that, I will finish. Thank you.
Russell, thank you very much. That was absolutely fascinating. I've got one question from Kerem that comes back to us, back to the sort of the seasonal aspect. How do humans adapt in seasons, and can we also switch off?
No, great question. We can't switch off as far as we can tell. But we do show altered patterns of sleep-wake across the seasons. What was interesting is, again, going back to that Ken Wright study, he did the first experiments during the summer, and then he did them in the winter. And he showed that sleep duration expanded a lot during the winter months. And there are other studies showing that, you can expand and contract your sleep-wake duration depending upon the season. So yes, to some extent it's masked, of course, because we don't get so much light exposure, but where there's evidence for it, you see it. Some lovely stuff that Helena Ilarova from the Czech Republic did some years ago, showing that agricultural workers would expand and contract their melatonin profile, beautifully. So we've got the equipment to detect seasons. And in the past, there's evidence that our behavior, was different. So the birth rate, for example, used to be much show a seasonal influence, but with industrialization. So some interesting studies have come out of Spain looking at the frequency of births pre-industrialization and then post-industrial. And you see that with industrialization, the seasonality of births has been really quite changed. Now, whether that's due to melatonin and whether we're behaving like sheep and deer, we don't know. It's very difficult to tease apart the reproductive. There are seasonal changes, but what's driving them, we don't know. There could be behavioral influences. So, for example, the Inuit and the northern peoples of Canada, birth records there showed that peak births until recently were all in March. And I think it was like 80% of births were in March. And then people suggested, well, that makes an awful lot of sense because March, it's the worst of the winter is over. You've got the spring, you've got food, it's reasonably climatic conditions, and you've got long enough to put on fat to survive the following winter. And then somebody bothered to ask the Inuit why they thought the maximum number of births were in around about March. And then they said, well, duh, that's because only time we have any privacy. We're not cooped up in a hut or an igloo. So we do show rhythms. And in fact, my book called Seasons of Life talks about this a lot. And one of the chapters in Light is on this. But the mechanisms underlying human seasonalities, and there's lots of it. So there are very strong, well, when I say strong, they are statistically significant rhythms in suicide, for example. You see rhythms in self-harm, and they're flipped from the northern to the southern hemisphere. They're 12 months, sorry, 6 months out of alignment. So there's something going on. What's driving it is unclear. And of course, one I think really interesting set of studies would be on the people of northern Canada, and what's going on with their circadian rhythms. But they've been abused so much, I think they've become very suspicious of any sorts of intervention. So it'd be very difficult to do those sorts of experiments.
So I've got a couple of questions from Kevin, if that's okay. So Kevin, thank you for the questions. Where can we get good information on the timing of regular medications, for blood pressure and diabetes control?
Yeah, I talk about this in the book A Lifetime, and it's fully referenced. So it's complicated. And it'll depend often about the half-life of the drug. So, for example, let me try and remember. If we think about aspirin, what aspirin does is abolish the stickiness of platelets. That's why, if you've got high blood pressure, for example, they often put on, it thins the blood. Now, aspirin only lasts a very short period of time, a couple of hours, effectively, in the blood. And the chances of a stroke are greatest between 6 a.m. and 12 noon. But, and that's associated with a sharp rise, a circadian-driven rise in blood pressure, but also increased blood viscosity, stickiness in that morning window. But the advice is that you take your aspirin before you go to sleep. So how the hell taking something before you go to sleep to interact with the real problem between 6 a.m. and 12 noon the next day? And the answer is because the platelets, and billions of them are manufactured, are made at night. So the aspirin is there to wipe out their stickiness. And so by the next morning, you have reduced plasma viscosity. So you might think the best time to take your aspirin would be in the morning because that's when the danger zone is, but actually it's the night before. So that's one example where it does get a bit confusing. There are about, as far as I can remember, 100 FDA approved drugs which do have a time of day effect. And Ulysses, if I can remember to do it, I'll try and dig out that paper and send it to you, and then you can get a better sense. I mean, again, part of the reason for writing a book was for people to ask their doctor, when should I take this? And of course, most of drug taking is compliance. So they often say, well, with a meal. But actually, that may not be the best time at all. And so the whole field of chronopharmacology is emerging, and people are paying more attention to it. The difficulty is, and sorry to go on, is that when you talk to the drug companies about this, they say, yeah, we know all about that. So we design drugs with a very long half-life. So we'll interact with the peak in the pathology. And then I sort of have to point out, well, you're giving a drug at a higher concentration than you need for a longer period than you need. And what are the consequences of that? Well, side effects. And so what do you do for side effects? You take more drugs. So they've been fairly lazy about optimizing drug taking, but it's a fascinating subject.
Amazing, thank you. So just one quick one. Flick, I saw the message. Yes, it's being recorded. And Russell, would you be happy to share your slides after
Yeah,
today?
sure.
Yes,
I mean,
brilliant.
fine.
Okay.
I'll turn them into a PDF and then send them to you.
That'd
Yeah.
be amazing. So are you okay for a few more moments?
Yeah,
Or
only I've
we
got to
tight?
leave pretty shortly because I'm picking up my wife and it's my turn to do dinner.
Amazing,
So,
amazing. So I'm
so,
going to wrap it up fairly quickly in
yeah.
that case. So Kevin had one more question, which was, what are your thoughts on infrared and near infrared impacts on mitochondria?
Oh, that's really interesting. I have some very distinctive views on that. So yes, I've looked at the papers which have shown that infrared exposure to athletes who've got strains and muscle things, and it does seem to be effective. Where I start to get less convinced is some of the proposed mechanisms. Now, uh, it's very easy to do these experiments because of the power of action spectroscopy, matching, the absorption spectrum of a pigment with the efficacy of different wavelengths of light. So the proposal is that it's cytochrome oxidase, I think, or cytochromes in the mitochondria that are absorbing the red light and changing the function of the mitochondria. If that's the proposal, then for God's sake, do an action spectrum. Because to my mind, it could just as easily be heat, which is what infrared, long wavelength light is, increasing blood flow, bringing more immune cells and more repair mechanisms to bear upon those tissues. So yes, I think red light is having an effect under certain circumstances. What are the mechanisms? I don't think we know yet, but the experiments are really simple to do. Well, relatively simple to do. Just do an action spectrum for the effects of red light. If it matches a cytochrome, then okay, I'm much more convinced it's interacting with the mitochondria.
So maybe one last question, if I may,
Sure.
before we let you run off to make dinner.
Make dinner,
What,
yeah.
what do you think would be a good place to start for architects, lighting designers, and other people in the building industry, building professionals, when designing healthier indoor environments?
Well, I feel that that last bullet point is the future is dialogue and research, and so I think if Comair do decide to bring the sector together, then I would want not just a bunch of scientists, but people in the commercial and the architecture and the lighting industry space. I think that would be it's critical to have that dialogue across the various sectors. And I'm very passionate about trying to make that happen if I'm in a position to do so.
Amazing. Thank you very much. Russell, I'm going to bring this one to a close. There's been lots of some very nice comments about how stimulating this conversation has been and your presentation. So thank you very much because it's been really inspiring. The series continues in November with Bob Fosbury and Professor Glenn Jeffrey, and we'll be talking there about light and light beyond photosynthesis and talking about mitochondrial infrared. Kevin, if you're happy to join in November, and then in December we'll be talking about melatonin, light, and the optics of the human body with Scott Zimmerman. Again, another hopefully fascinating talk. But Russell, go and make a wonderful dinner for your wife. Thank you so much for all your time because we are over time, but it has been absolutely fascinating and really appreciate your time.
Well, thank you, Eunice, for the opportunity, and I wish everybody a pleasant evening. So thanks again. Take care.
Thank you very
Bye-bye.
much. Good night.
Bye.
Take care.